Hey, in this video we're going to talk about lipids. If you remember from last video, we talked about carbohydrates specifically, and so this one's going to mirror that video in that we're going to talk about the ways in which the components bond in order to make the organic molecule known as lipids. But in this one, like I said, we're going to focus on just the organic molecule lipids. Now, lipids come in a variety of different structures, but the ones that we're going to focus on for the purposes of the IB syllabus are really in phospholipids and triglyceride. And so let's take a little time discussing what is required in order for a lipid to be a phospholipid versus a triglyceride and what the functions are for each. Remember, this is another case of structure dictates function, and so these two lipids, while they look similar, are structured very differently because they are used in entirely different situations. Okay, and so if we focus on phospholipid, which is the structure on the left, a phospholipid is made up of the same structures or the same chemicals that a triglyceride, in that you will see a glycerol, which is found here, and specifically you could say even here, and it's made of fatty acid chains. Okay, these fatty acids are repeating CH2s. And so this would be a carbon, 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 carbon all the way down. Notice that in some of these fatty acids, you notice that there's a double bond, which makes it unsaturated. We're going to talk about unsaturated versus saturated in a little bit. But for now, notice that two fatty acid chains make up or constitute a phospholipid. Okay? And what makes a phospholipid, and what gives the phospholipid its proprietary property, if you will, is this phosphate head. Okay, phosphate head. Notice that when you look at the entire molecule, it looks like it has a head with two chains or two tails. I mean, it's almost like a balloon with two strings. Okay, what is the purpose of a phospholipid? Phospholipids are going to orient themselves in a phospholipid bilayer, which we will talk about later in cell biology or cytology, but this structure is perfectly designed in order to produce a bilayer that is going to give all cells the ability to maintain a boundary between internal and outside of the cell, which is vital to not only structuring the cell but also maintaining life as we know it. Because there is a phosphate in the head of this molecule, the head has a hydrophilic property, so we call the head hydrophilic. We also say, or can say, that the head is polar, and so it is going to interact well with water. And the tails, being lipid in nature, they are hydrophobic, and so they are non-polar. And because they are non-polar, they are going to not mix with water very well. Okay, in fact, they're going to be insoluble. They're going to repel water. They're not going to orient themselves towards water, which is why phospholipid tails are going to create this membrane where tails are pointed inward and heads are pointed outward. That way, H2O, which is obviously in our environment and outside of the cell, and H2O, which is obviously a main component of the cell, can be kept outside of this zone of hydrophobicity, if you will. Okay, zone of hydrophobicity, which means hydrophobic zone. So there is absolutely no water until you get into membrane transport, which we will in later videos, in which water has to go through membranes, but it uses or utilizes special proteins, alkylporins, which we'll talk about at the time. Okay, so for the purposes of this video and the purposes of this particular section, specifically 2.3 in IB syllabus, or in the new IB syllabus, the phospholipid is made up of essentially two things, or I would say essentially three things: a phosphate, a glycerol, and two fatty acid chains. Okay, fatty acid chains are hydrophobic and nonpolar. The head, which is made up of the glycerol and phosphate, is polar and hydrophilic. On the other side is a triglyceride, and these triglycerides are entirely nonpolar. Okay, which means they are hydrophobic, okay, which means they do not interact with water very well. Okay, in fact, they're insoluble, And you can simply demonstrate this by pouring vegetable oil or any kind of oil into water. Oil is made up of these molecules, triglycerides, and so you will notice when you do that demonstration that the oil wants to separate itself. Now, it will separate itself based on density as well. Obviously, oil is less dense than water, which is why it floats, but there is a very distinct boundary between the oil and the water. There is no mixing of the two. And because there is no mixing and or no dissolving, it is directly related or directly due to the fact that oil is hydrophobic because of these triglycerides. Okay, you'll notice that it has a glycerol backbone, and we can also say it kind of has a glycerol head. Okay, this is the glycerol, and the glycerol is the exact same as this structure. Notice I put a box around this glycerol, and I put a box around this glycerol. Glycerol constitutes the head of a triglyceride, but triglycerides have 3 fatty acid chains and no phosphate. Okay, no phosphate and 3 fatty acid chains. Now, what is the purpose of a triglyceride? We already said the purpose of a phospholipid was to create these boundaries between cells, orient themselves in a way that is going to allow for this double membrane or double lipid bilayer structure to form. Triglycerides are not involved in membrane construction. They are actually going to store energy long term, and so they are an energy storage molecule, and it makes sense that if I say that energy is stored in fatty acids, then obviously a triglyceride is going to be more structured or is going to be better structured to allow more energy to be stored in every molecule, okay? Because triglyceride, because it has an extra fatty acid than a phospholipid, notice there's 2 over here and 3 over here, that makes a triglyceride. For every triglyceride, there is an extra 50%, okay? There's 50% more energy storage potential in a triglyceride than there would be in a phospholipid. So you don't have to store as many triglycerides to actually store more energy. And again, that's just one example of structure dictates function. These molecules right here are structured in a way that is going to allow them to store energy long term. And these molecules in phospholipids are actually going to structure themselves and have structured themselves to allow for bilayers to form and therefore membranes to form. Okay, if we focus on triglycerides, like I just talked about, they have 3 fatty acids, and they are linked to the glycerol backbone or linked to the glycerol head by ester bonds. Okay, now how do the fatty acids actually bond to the glycerol? You can see right here. It is the same kind of condensation reaction we've been talking about in the last 2 videos. We are going to take an H from the glycerol. Okay, we're going to take an H from one of the OHs on the glycerol. We're going to take an OH from the carboxyl end of a fatty acid, and because we have an OH and an H, we are going to produce H2O, which is a condensation reaction, right? It's a type of anabolic reaction, and because we remove H2O, this no longer exists on this carboxyl, and so this carbon is going to create a bond with the oxygen, which you're going to see right here. Okay, there's the carbon to oxygen bond. That's what's holding the fatty acid to the glycerol. It is a covalent bond, or I should say it's a type of covalent bond. Remember, covalent bonds are weak, but it is a specific type of covalent bond, an ester bond. Okay, so we have 3 ester bonds that are holding the 3 fatty acids to the glycerol backbone. And because we linked 3 fatty acids to the glycerol, we have released 3 water molecules per triglyceride that is produced. And like I said in the last slide, the triglycerides are perfectly structured to allow them to store energy long term in both plants and animals. Now, fats are obviously stored in animals, and oils are stored in plants. How do you know? When you go to the store and you look at the vegetable oil aisle and you see all the different varieties of oils like canola oil and safflower seed oil and olive oil and even vegetable oil, those are all plant derived. Fats, on the other hand, are not something that you typically see or experience when cooking plants. When you cook meat products like ground beef or bacon or something of that nature, you typically have a bunch of fat that has rendered off of the meat that you're cooking because it is animal in nature. So fats are triglycerides in animals, oils are triglycerides in plants, both of which have the exact same function and have the exact same structure. It just depends on the origin of the molecule to basically the triglyceride unsaturation level, which we'll get to later, depends on where it's derived from. So oils are going to be, for the most part, unsaturated, and fats are going to be, for the most part, saturated, which will make more sense in a minute. Okay, so I just talked about saturated versus unsaturated, and so now we need to talk about what the structure is that relates to that particular name. Okay, and so you'll notice that saturated and unsaturated are the two distinct classes of fatty acids. Now, when something is typically saturated, like a piece of clothing being saturated with water, for instance, it would mean that it is completely full of oil or completely full of water. And if something is unsaturated, it is not completely full. Okay, so saturated. When we refer to a fatty acid as being saturated, we are talking about it being completely full of hydrogen. Okay, notice that every single one of these carbons is completely saturated with hydrogens. There are no double bonds in a saturated, okay? No double bonds in a saturated fatty acid. On the other side, unsaturated means that it is not completely full, okay? And so it means that there are double bonds in an unsaturated fatty acid. Now, the degree to which it is unsaturated is dependent on the number of double bonds, okay? So as the double bonds increase, then the level of unsaturation increases as well. Okay, so the more double bonds, the more unsaturated the fatty acid is. The fewer the double bonds, the less unsaturated fatty acid, or the less unsaturated it is. And if there are no double bonds at all, it is completely saturated or full of hydrogens. Okay, so what does that typically mean, or what does it look like? These are all examples of fatty acids and what the structure looks like and what the number of double bonds do to that particular fatty acid. This right here would be an example of a monounsaturated fatty acid because there is a single double bond. Double bond is right here. Notice that when the double bond happens, the resulting chain bends. Okay, so double bonds are going to elicit a bend or a kink in the fatty acid. The more double bonds, the more bendable and the more flexible the fatty acid gets. So as unsaturation increases, the amount of flexibility increases as well. It's directly proportional. Okay, and as the level of unsaturation decreases, okay, I'm just going to simplify this. As the unsaturation decreases, then the flexibility decreases as well. Okay, and then you get to a completely saturated fatty acid. This is very rigid. Okay, it is not flexible at all. What are the different types of unsaturated? You have a saturated, okay, which means that your carbons are completely full of hydrogen, which can be seen here. Okay, can also be seen here. This one is a saturated. This one is a saturated. This one is a unsaturated because of the double bond you see here. And because of the double bond, it forms a bend in this particular fatty acid right there. Now down here, saturated, completely saturated with hydrogen. Monounsaturated means that you have a single. Mono means one. You have a single double bond Poly, just like polypeptide or polysaccharide, you have multiple, 1, 2, and you could have as many as 3, 4, 5, 6, 7. I mean, as many double bonds results in a more unsaturated fatty acid. Okay? One thing, though, to notice from all of these diagrams, because it's a very good likelihood, there's a good chance you're going to be given a diagram or a specific chemical like this on a test, unlabeled, and you're going to have to identify it based on some characteristics that you should know or that you should be familiar with from carbohydrates and lipids and carbohydrates, lipids, proteins, and nucleic acids, and that every single lipid that you will ever see on an IB exam or on any kind of biology literature or whatever is, you should notice this COOH group. COOH group. Remember, that is the carboxyl group. The COOH group is the group that you can usually identify a lipid with. Okay, COOH, COOH, COOH. All of these have that COOH. That is their carboxyl. Like I said, all lipids have it. Now let's talk a little bit about the difference between cis and trans in terms of the orientation of the particular molecule. In a saturated fatty acid, remember, or a saturated fat, you're going to have completely saturated the carbons with hydrogen. So there are no double bonds, and so it is completely saturated and it remains rigid. Okay? In an unsaturated fat, you can have two choices. You either have a trans configuration or you have a cis configuration. When we talk about diets, I know it has become popular to look for trans fat, and in fact trans fat is typically one of those things found on a nutrition label. Unsaturated fat is also on, well, saturated, unsaturated, and trans fat are all on those food labels, but the difference between a trans fat and a unsaturated fat is the configuration. Trans fat are called trans because they have a trans configuration, and unsaturated fats typically have a cis configuration. So what that means is both of these are unsaturated, meaning they have a double bond, but depending on which side of the carbons that are in this double bond the hydrogens are on, indicates the configuration of that particular unsaturated fat. So cis, I remember cis, I know it starts with a C, but it kind of sounds like the S sound, and so cis is same. Okay, cis same. Cis means that the hydrogens are on the same side of the carbons, and so if the carbon has the hydrogen on top and it's the same way in both cases, or both carbons that are interacting in the same double bond, then it is a cis configuration, and it's going to form a kink or a bend. Okay, it's going to form a bend. It is less rigid, or it is more flexible. Trans fat, trans means that it's like transverse, right? You have one hydrogen on top, one hydrogen on the bottom. They are on opposite sides, and so trans is the opposite. And because they are on the opposite, they are not going to form a bend. Okay, no bend, and because there's no bend, there's no flexibility. In fact, it's just like being saturated. And so if you look at this diagram over here, which we can blow up, cis configuration, the same side, hydrogens are on the same side, that is a cis double bond. It forms a bend, and so this one becomes flexible. That's good for you. Here, because the hydrogen is on opposite sides, it locks it in, it stays rigid, and it is not flexible. That is bad for you. This is just like a saturated fatty acid. Okay, it acts like a saturated fatty acid because it is not flexible. Okay, what does that mean? It just means they're less healthy. Okay, trans fats are not as healthy as the unsaturated fats. Okay, are more healthy. Why are they healthy? They stay liquid at room temp, which includes body temp. Okay, it stays liquid at body temp. The solid at room temp is a really bad property because it could be solid at body temp as well. So these are the types of fats. The unsaturated fats and the trans fats are bad because they will often solidify in your body temperature, which means they can lead to plaque formation in your arteries, lead to plaque formation in vessel walls and artery walls. These are typically artificially synthesized, and it's used or synthesized in a production called hydrogenation. Okay, basically like your margarines, fake butters, stuff like that have trans fats. They are artificially synthesized. They stay solid at room temp. These unsaturated fats, because they have these bends or kinks, are more flexible, and because they're more flexible, they are not as rigid, and so they stay liquid at room temp. These are your things like oils, canola oil, sunflower seed oil, olive oil, stuff like that. And so if we use our understanding of what these cis and trans double bonds do to a particular fatty acid, we need to now kind of test our understanding by looking at specific molecules, specific fatty acids that may pop themselves up in IB questions as we go through our IB syllabus. Okay, and so if you look at these fatty acids, you're going to see that there are different shapes. We know that every single one of these is a fatty acid, most noticeably by the COOH group. Okay, this would be COOH, carboxyl. That's a carboxyl group. This is our carboxyl group. This is a carboxyl group, COOH, COOH. And so it doesn't matter if it's written out as COOH or if it is written out as an OOH. You know that this last little bond right here, this last little bend angle, if you will, is a carbon on these three examples. So we have 4 different fatty acids in this particular slide, and all of them look different. All of them have a different structure, a different shape, but we need to identify not only that these 4 are in fact fatty acids, but we need to potentially identify if the double bond, which can be seen here and here and here, and several different places, okay, double bonds everywhere, we need to recognize if these double bonds are cis or trans configurations specifically. And so this one, because the hydrogen is on the same side, this would be a carbon, 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 but because the hydrogen would be off here and here, we know that they're on the same side, and so that is a cis configuration. Same with this one, cis, 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 and so on. Every single one of the double bonds in this particular fatty acid is a cis configuration, because you know that if you were to look at the hydrogen would be on the same side of these double bonds in every one of these particular cases. Okay, if we go down here to the bottom right, we know that this is a cis configuration because our hydrogens would be located on the same side. However, on this example, notice that the carbon, which is here and here, would have a different hydrogen configuration, and so this one is a trans configuration, the only trans configuration on this entire slide. Okay, so 4 examples. Most of the examples on this particular slide of these examples would be cis configuration. There is one, however, that is a trans configuration, and so you can see how it's different and how it looks differently than all the other cis configurations. Notice that the cis configurations provide this fatty acid to be a lot more flexible. In fact, if you look at the overall shape, it is a lot more flexible and less rigid than these, which have basically just a long rigid chain. This one is more unsaturated. Okay, the level or the degree of unsaturation is increased. Okay, unsaturation is increased, and so it is more flexible than these examples which only have one. Again, if we bring in more understanding or more vocabulary, this would be a monounsaturated, because there's a single double bond. This would be a monounsaturated. This would be a, well, these two over here would be polyunsaturated. Okay, polyunsaturated is basically 2 or more. This is a polyunsaturated. This is a polyunsaturated, but this one is more unsaturated than this one. So these two are the least unsaturated. This would be the next most unsaturated. This would be the most unsaturated. Okay? So, a couple questions to kind of end this discussion. Why are lipids more suitable for storing long-term energy in humans than carbohydrates? Remember, carbohydrates and lipids both store energy, but carbohydrates store short-term energy while lipids store long-term energy. And so, why are lipids more suitable for storing long-term energy than carbohydrates? And it has to do with the long chains of hydrocarbons, which is the energy storage component to the triglycerides, and so that is the fatty acids. Okay, the fact that triglycerides are kept in this layer of adipose tissue is really important for the conservation of energy in the long run, because if we can insulate the body for warmth, then we're not going to use excess energy keeping the body warm by burning through calories or burning through unneeded energy or spending unneeded energy on conserving the body's temperature if we can just insulate it in the first place. Adipose tissue is also important when we talk about cushioning organs from potential energy. Okay, we not only keep adipose tissue for long-term energy storage, we keep adipose tissue for insulation to keep us warm or to conserve energy in the long term, but we also use it in between organs in order to cushion them to prevent energy. Okay, and we have sphingolipids, which are really, really important when we talk about neuron transmission or electrical impulse, which we're going to talk about later on in the syllabus in future videos. But there is a sheath called myelin sheath. Myelinization is a process of actually surrounding the neurons with a type of insulating triglyceride or a insulating lipid called sphingolipids, which are going to be used to insulate those neurons so that we don't lose signal as we transmit those electrical impulses from neuron to neuron. Okay, that's really important when we talk about motor control, motor functioning, getting an impulse from brain to ultimately the muscle in which it needs to act on in order to move the body or transmit relay signals from brain to structure. Phospholipids are the other type of lipid, and so they are utilized as the main component to cellular membranes, and that's really important when we talk about maintaining boundary between inside and outside of the cell. And steroids are lipid-based, and they are involved in communication throughout the body and between cells. They are really, really important when we talk about cell signaling, which all of these lipids that play into humans' ability or animals, organisms' ability to store energy long term is really, really important. Okay, structure dictates function. It's really, really important for you to understand that lipids are structured in a way, and there are various different structures of lipids, but they are structured in a way that allows them to function a certain way. So let's look at this concept in a more picture-based approach. When we talk about why, or the question, or we question why lipids are structured the way they are and why that structure is more suitable for storing energy long term, it really comes down to two different concepts. One is they are lighter than carbohydrates. They equal or equate to less body mass, and they produce more energy. So you're able to keep the organism body mass or the organismal weight to a minimum while maximizing the amount of energy potential that organism has. And so if you are lighter, you have less energy requirements in order to maintain normal body functioning. You have less energy expenditure when you're talking about moving the organism. You have less cellular demand in terms of cellular metabolism. And so it just requires fewer nutrients, fewer energies produced to energize that amount of body mass. And so because lipids are 1/6 the weight or 1/6 the mass as carbohydrates, they are much, much lighter, and we can keep organisms lighter, less massful, or less massive, and that's really, really important when we talk about cellular metabolism and the energy requirements or energy demands placed on each and every cell. It's really, really important to look at this in terms of kind of minimizing mass. Okay, we want to minimize mass while maximizing energy. Okay, the less mass we have, the less cellular demands per cell, and and then when you pair that with a molecule that is more energetic or more energy potential or has more energy potential, it's a win-win. 1 gram of glycogen, which is a polysaccharide. Remember, glycogen is the carbohydrate or the polymer form of carbohydrate that animals use, and that includes humans. Okay, glycogen is the way we as humans store energy short term. It is a polysaccharide, and if we stored the energy that we would need, that lipids afford us, in the form of glycogen, you would have to be 6 times more massive than you currently are, which is going to decrease the efficiency of every one of your cells, and it's going to require 6 times more energy just to break even when we talk about cellular demands. Okay, and so it's really, really beneficial from an organism standpoint to be less massive and simultaneously more energetic, which is essentially the benefit that lipids afford us. The amount of energy is 2 times more energetic in a gram of lipid than a gram of carbohydrates. So while we are simultaneously 1/6 the mass when we talk about lipids, okay, lipids are 1/6 the mass, but afford us 2 times the amount of energy. It's a win-win. You want to be less massive and you want to provide as much energy, and so this is kind of a way to look at that. The energy in 1 gram of lipids divided by energy of 2 grams per carbon plus 4 grams of water would be kind of a simple equation used to calculate the total mass needed to store the energy that we require to live. Okay? Again, lipids are better than glycogen. We just talked about lipids being less massive and providing us 2 times as much energy. And the benefit, though, of glycogen and the reason why we do need glycogen, remember glycogen is a polysaccharide that we absolutely can't live without. It doesn't afford us long-term energy storage, but it does provide a short-term energy storage, and the reason it is short-term is that the glucose molecules, remember there are glucose rings that are bonded together in a network of monosaccharides. Okay, remember they're linked by condensation reaction, but each one of these is a glucose molecule. These glucose molecules can be readily broken off as needed in order to be transported or pumped to the blood to raise the ATP production per cell, which ultimately provides energy to that particular cell. So we need glycogen so that we readily can break them off or break them off at will as needed to produce energy at the cellular level. Triglycerides are broken down very slowly, again, which is why it's long-term energy storage. We store energy as triglycerides, as fats and oils, for the long term. This is our kind of long-term supply. We need those in case we have long periods of time when food isn't available, we're stranded somewhere, we don't have access to food. We need to break them down very slowly to support life for the long term. Okay? Glucose is the energy currency for the cell and is required for cellular energetics, specifically cell respiration, to produce adenosine triphosphate, which I talked about earlier. Okay? Glucose can be converted to ATP. It can be stored as glycogen in the liver or be converted to fat, adipose tissue. It really depends on what the glucose demand is in the cell. If there is plenty of glucose in the blood already, it's going to be stored as glycogen in the cell or in the liver. If there is plenty of glycogen and or there is way too much to be used or there's way too much for an extended period of time, it will readily convert that sugar into adipose tissue to make sure we have adequate fat reserves for the long term. Okay? We're going to finish the lecture with a little analogy. I think this just kind of puts it in perspective, makes it very easy as a flow chart to kind of understand where we're going. But if you are paid in cash, okay, and I know most people work for a paycheck and or direct deposit nowadays, but if you were paid in cash, you really have 3 options. We're going to use cash as our analogy, but cash in this example would be analogous to glucose. Okay, so you're paid in cash. 3 examples. You can either pocket the money and or put it in your wallet, right? You're going to wallet your money, put it in your pocket. You can deposit it in the bank for long term. Not as easily accessible, but you can store it long term. It's safer, right? Or you can spend it. Glucose is immediately spent or utilized in cell respiration as ATP, right? So if glucose is going to be spent, then it's going to be produced into ATP. This ATP is going to be quickly utilized as an energy source to keep the cell going. If you are to put it in your wallet, it is very easy to get to. You can get to it at will. You don't have to wait and drive to the bank. You don't have to wait and put a withdrawal to get your own money. It is very easily accessible and easy to get to. You carry your wallet on you all the time. Now, is it possible to keep in your wallet every dollar you have to your name? I would hope not, because if you did, then it would be too large. Your wallet would literally be too large for you to carry all the time. There isn't enough space in your wallet to support the weight and or mass of all of the dollar bills or all of the cash, and so you cannot carry every bit of money that you have in savings, right? It can only hold so much. It's a definitive size or a definite size, and so we have bank accounts that are going to be a deposit site for the money that we can't or don't want to carry. And you can put in virtually unlimited amount of money here, but it's more of a hassle and it's a slower process to get it out. Okay, so this is very easy to get to. It's very fast. This is a slower process to get your money. It's a safer process, right? Your money is safer in a bank than it would be on you. But if this was an analogy, like I said, glucose can either be spent, utilized as ATP, or it can be stored short term at small amounts in a way that makes it easy to get to, very quickly get to it, or it can be deposited for the long term. That would be adipose tissue or the fat, but it is a slower process to get it out. Okay, so that's where we're going to end. Bring your questions to class. See ya.